Now, next, we'd like to invite our speaker from CSIR CDRI, Dr. Shrikant Kumar Rath. He is a very accomplished scientist and very well published researcher. For past few decades, he has been involved in the field of genotoxicity, and he is currently working on genomics and cancer research and understanding the principles behind the toxicity in these model systems. And I humbly invite Dr. Rath for his talk in this platform.
Dr.
Sir,
Rath, are you there? Yeah.
Yeah,
he's there.
I'm here.
Yeah, we can see your screen. Just go to slideshow and
Yeah, I am going. Can you see my slides?
yes, 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 sir.
Yeah, good afternoon, everybody. I am very grateful to Professor Monisha Banerjee and her associates, Dr. Malini, Dr. Asutosh, Dr. Amita, Dr. Poonam, for organizing such a exciting conference webinar. As for the first time, actually, I'm attending a webinar, sitting from my bedroom and talking to so many people. Around 150 people are attending now. I shall describe about what the biologist research scholars should do and how I will go to the basics, then the actually highly extended talks by my previous lectures. I would like to focus on the students' characters, how they can address the biology research from their own labs. Having said that, I first ask this question: why we need to do research? Is it actually required? Are we required to do research? Without research, can we not survive? Yes, we can survive, but just imagine if you go back to 100 years, then you can understand what are the facilities we had previously, and they are not available there. What is available today? For example, we are in our school days, we are driving to a place by cycles, but today we have all metro trains, buses, turbo buses, all types of facilities are there. All it came from lot of research inputs. Lot of research have been done in varieties of it, not in biology, but for everything. But we biologists are also very responsible. We should also do a responsible work to contribute to this science. Why? Because we are the human being is the only savior. Only it can take care of the climate. Only human being can take its environment to take care. Because we are all dependent upon all the environment and ecosystems. We require something from, we derive something from our ecosystem, and the ecosystem also derives pleasure if we are taking care of them. For example, if we are talking of in ecosystem, we have viruses, we have bacteria, we have different animals, we have different types of plants. And if you ask a question that whether we have actually identified all of them, it is certainly no. We might have done some research about some species. We might not have done some research about some other species. Some of the species of plants and animals in Himalaya may not be known to us. What is known to us today are actually very few species which are actually around our environment. For example, if you do very good research, for example, you are doing adenovirus, for example, is used in our technology where we require vaccines. We use adenovirus for transgenic technologies. Almost all transgenic technologies, they are trying with adenovirus just to put a transgene inside the inside the genome of a host, then try to manipulate the host to derive the best out of it, so that it can be beneficial for the human race. Similarly, if we are talking of fish, we are trying to making transgenic fishes, which are actually for fishes are very important as well, for food as well as ornamental. Small ornamental fishes are being people are making just to sell it in the market, just to get some economy. At the same time, the other people, they try to do the fishes with growth hormones genes, so that the fish at the younger age can be very big, so that the more money can be earned out of that. Similarly, we have several plants which can actually give more oxygens and can be actually planted inside the house, so that we can get more oxygen while staying in the home, so that we don't have to struggle for the oxygen in our daily life. For example, deers, cows, all these animals. For example, deers, in different zoos you can find out beautiful deers in different straps, different colors, just because we have taken actual breeding activities in different combinations, so that we can get them in the zoo, so that people who can visit the zoo can get them entertained and enjoyed. Similarly, if we are talking of cows and buffaloes, if we are getting more milk from the cows, so best variety of breeding we do and get the, all these comes in the biology. And different tools and techniques have been utilized, where it is from the classical breeding to the transgenic technologies. Everything we use to get the best breeds for the benefits of the society. At the same time, we should not forget that we have to take care of the ecosystem, so that we should not disturb the ecosystem, and the ecosystem and the people can actually live for longer, not for only 100 years, but uh, for generations to come, for generation to come, for thousands year or thousands of years, so that our next generations can enjoy their life on this earth. Taking to who does research, can it is a platter for everybody? No, we everybody cannot do research, because research scholar has got a quality. So research scholars who have joined actually newly research, they should understand that why they, how they can manipulate themselves, so that they can be a good researchers. We have generally in India, due to our economic conditions, people are more attracted towards the job like IAS, IPS, administrative services, because the job requirement is very different. You have power to execute, you have money to enjoy, but a researcher at the same time in the same age doesn't have anything. But one thing is very good for researcher is a uh, man who is full of freedom, full of wisdom, full free to do anything. The researcher can sell his idea and get funded by big funding agencies to pursue his research. So that is the big difference. Crores of rupees can be funded to you to do your research. You may not get anything at the end of the day, but you are actually free to spend them. You are free to move around the world. You can interact with the people. You can go and deliver your lectures, your experience, and get derive the knowledge. So researcher is a very unique species that you have to understand. And I would like to congratulate all of you who are doing good research. You will do good research. It may be, there is no good or bad research. Research is research, but always research is good. If you are not doing very good research, then it is better not to do research again. So you have to have an interest in the subject. If you do not have an interest, the interest has to be like you love your mother. In the same love, you have to do the subject. You have to do the justice to the subject, so that the subject can be improved by your research. You have to fall in love with the subject and extensively read the subjects, so that you can actually execute knowledge and implement the knowledge to do further research. So sound knowledge is must, with a good love to the towards the subject. Then you have to be very determined and dedication. If you are not determined, then you cannot do actually research. Absolutely, you cannot do that, because you will always be, you will be not a confident person. If you are not confident, then whatever you are doing in research may not get the absolute justice, because if you are not determined, you cannot take a decision what is, whether you, whatever you are observing is right or wrong. So that determination has to be there, and when there is determination, you have interest, then full dedication will be there, so that you can full time you can give to research. I know you are a human being like any others. You have some many other jobs to do, but the research has to be uniquely treated, and your dedication to be to the subject has to be there. You have to have honesty. Yeah, now it is okay. Yeah. So let me go back to the slides again. Who does the research? One should have very lot of interest in the subject, and you have to fall in love with the subject extensively. You should read so that you can gain knowledge, and you have to have sound knowledge in the subject so that you can further plan your research, and you have to have a determination and dedication for the subjects. Then only you can properly do the research with all lot of curiosity, honesty, and integrity. As I was talking about, recently there is a lot of discussion about the pop peer. People have cited never cared of. So one has to think of it very in advance, so that the research cannot be challenged like that. The extra knowledge can be added, but it should not be what they have done is wrong. So one should not talk about that. So for that you require a sincerity and a lot of patience. Without patience, you cannot do research, because for the parallelly you must be watching your friends are getting promoted in the administration and doing many things, and at the same time you are moving around in a university campus who as a students. So sometimes it, hello, you have to be very interpret your results appropriately, so that, hello, you have to interpret your result appropriately with an excellent communication. A researcher has to be excellent communicator, otherwise he cannot go further. For example, the challenges in biology is many. The starting from the virus to the coronavirus, from to the different species of human beings, there are very wide variety of subjects to study. From the submicroscopic organisms like poliovirus, tobacco mosaic virus, Sendai virus, coronavirus, to the humans of 15 species. Many may not be knowing that we have 15 species and only one species is surviving. What is the cause why 14 species have gone from the earth? And at the same time, there are several ideal species which can be utilized in the laboratory to study different types of physiology, cell biology, molecular biology tools we can use, and we study different, starting from neurobiology to developmental biology. We can use by using this model systems like C. elegans, Drosophila, fish, birds, dogs, elephants, and elephants, for example, African elephants and Indian elephants, why they are very different, how we can distinguish between from the far off. And those who are pet lovers, they must be knowing the variety of breeds we have. And people are doing research, lot of research to do the aggressiveness, research on the aggressiveness, behavior, their qualities of sniffing and other things. So a lot of, it's a variety of studies one can do as a biologist in a research. So the types are, you have to, many types, you can find out the types. For example, I have given the example of Himalayan algae or fungi or bacteria are very different what is available in the Kanyakumari, and that is because of the adaptations and other things. But exactly are we there to know that how many types we have already observed, how many types we have already recorded, how many types, how they are very different from one another, how they have adapted, how their physiology is working, how their biochemical pathways which are different from the humans or similar to that, or their behavior is very, very different in different conditions and salinity, pH or any other conditions. Their locomotions, are they different or they are same, or their behavior in a disease conditions. So your aim and objectives decides your methods to be followed. So methods are very, very different. If you want to study a microscopic individual, for example, a bacterium, so you require electron microscope, you require a small microscope, you require different types of molecular biological tools to understand what are the proteins and peptides it can secrete. You require the genomic research tools to find out their genome size, and you require what are the genes they are getting expressed in what conditions. For example, your approaches, your experimental tools, your statistical tools, all you, and what is the common? And the common is you and your guide. So always there should be a good interaction with the guide and you, then only you can pursue all these things. And mostly in biological students, they used to get rid of statistical tools. They used to not to read the statistics. They don't understand what is biostatistics. And as a researcher, you cannot actually get out of statistics. You have to implement statistics. You have to know the statistics, and you have to know what is the appropriate statistical method you should use to analyze your data, because different types of data requires different types of statistical tools to pursue. Similarly, how to select the research topic, it depends upon the research interest, funding, facility, feasibility, and researcher's willpower. A researcher's willpower is the first requirement. If you can do that, if you have a willpower, strong willpower, then you can actually manage to do everything. You are very, very intelligent, then you can get a good guide, you can get your research funded, you can work in a better facility, or you can work together with other workers in different facilities and can make your research feasible. So every research is relevant. You have to make it contemporary. If some people they talk about I will do only in cancer research, I want to do in the molecular biology. Molecular biology is a tool, but starting from classical to the experimental tools, you have to actually make it contemporary. So research scholar is the most important thing in the whole area of research.
कहाँ से चली गई आई नहीं हूँ
Most important thing for a research scholar is reading. Nowadays, students are not reading what actually it is expected to from them. They are not reading. They are not reading. Frankly speaking, I have been working in the research area for last 30 years, and I am finding reading quality has gone down because of maybe computer, maybe soft skills available, soft computer, soft copies are available. People are not able to read it properly. And that is the problem actually I wanted to address. I wanted to advise all the students to do that fast. Select papers from reputed journals. I am not telling that reputed journals means you go high impact factor journals, but you also consider the best society journals. You read Indian journals, you read foreign journals, you read Nature, Science, PNS, where the good papers are coming. And then, of course, there are many predatory journals you don't, please, I will advise you don't follow them. Then how to read faster thousands of papers, that is a challenge, and I wish to give a trick to you all. The best trick is to read the title, and title must ignite the progressive thinking process in you. If it is not igniting a progressive thinking process you, then it is not certainly the paper for you. So that is most important. You have to read the title and think. Just close your eyes and think about the what would be the description points in this paper. If it is igniting the progressive thinking process, then this is the paper you have to read. And if the paper ignites progressive thinking process, then read about the abstract, and you will realize the abstract must match your expectation. If not, stop for the reading. If it is, then it matches, then go further to read the questions the authors ask, tools they have utilized, and the results they obtained. Make a note of references card for future use. In one of my classes, I asked the student, do you know what is reference card? They do not know it. But the very first, I remember the very first meeting with my guide is I got a bunch of reference cards, and he asked me to go to the library to utilize these reference cards. Reference cards are very, very useful. Nowadays, what EndNote and other things are actually doing in computer system, reference cards we used to do. We used to write the reference and exactly the focus of the paper, so that during the writing of the thesis or during write of the paper, these reference cards are being utilized to cite the cite the paper in your research paper. So, so before starting any research, you require a plan. Okay, so plan is nothing but you have to discuss extensively after reading to with your guide, and after that you make a hypothesis. Then you go actually about a plan. A good plan is work half done. So most of the students nowadays, they do not have a plan in writing. They have maybe a plan, they must be a plan inside his brain, but they do not write it on a paper and pen. So one has to, I will advise, they should have a good plan, which is has to be a workable work plan, feasible work plan, and a good plan is work have done, and then perform carefully. Execution is highly needed. If you perform carefully, then actually most of your problems are being solved. Then you have to record everything. If you go, we Indians are very, very poor in recording. Let me tell you very frankly, there are a few years back, there is 2-3 years back, there is a actually a research paper we are talking of. The Japanese are very, very good researcher. They write everything on the notebook, but we Indians are very, very poor. You go and ask your students, they themselves can check every day what they do in the research lab. They do not write it. They always carry their research notebook on their brain, and they think that they will remember every time everything. So that is not possible. In my principle, if it is not recorded, it is not done. So therefore, you have to record every day. You have to record everything in details. If you have taken a 1 microliter solution and added 10 microliter of another solution, it has to be written carefully and it has to be followed carefully. Then it has to be audited carefully. Guide has to take care of this. To time, it has to audit, so that you can actually monitor the student, how he is going, what direction it is going, whether he is interpreting the result properly or not. If it is not, at the end of the day, in the 4 years end, if you guide, you show the record to guide, he may or she may get confused. So therefore, you have to have this auditing system in the place every month, every week. That will be a very good thing. Nowadays, DAC is there, so it is always better to present in front of the DAC, so that you can also improve your speaking power in addition to your research activities. Then accurate reporting is must. For example, in a group of animals of 10, if 8 are showing some types of trend and 2 are not, then there are several questions why these 2 are not showing the same thing. Whether the animals are of the same age, or they are actually of the same sex, or the animals are outliers. You have to decide in that point of time. You have to accurately report that why these 2 are not responding, and you have to have an explanation for that, and you have to find explanation for that. Then preserve for future references. You have to archive all your data. Even if you have completed PhD 4 years back, your record has to be retrieved from your lab after the 4 years, because sometimes things may require your data to be revisited, and that is a very good thing, so that one has to preserve the data. Nowadays, students only write everything on the computer, and while going, they take the laptop, and the guide is actually without any data when the student she or he leaves. So that is not a good practice. One has to have a record, positively hard bound records in the lab, so that if the guide wants to refer it, then it can or she can he or she can actually refer it in a right direction. How research becomes relevant? Many a time the students ask, I am not doing a very good science like you, sir. Many a time students develop complex that they are not working in the frontline areas. It is not like that actually. Research is research, and every area is actually important. For example, let us discuss about the corona disease, COVID-19. We do not know. We were not knowing before November 2019. Nobody was knowing what the COVID is going to do a havoc. We are only watching in around December or January that in China something happened and some virus is killing. But slowly you just see the first case of corona came to Kerala in 30th January, and slowly it has now killed around 7 lakh infected people, and around 20,000 people have died in India, and globally 113 lakh infected, affecting around 215 countries. Then the percentage of global rate death is 8%, and Indian death rate is 2.8. Then there is a question actually, how Indians are very different than the India. In India, only 2.8%, while globally it is 8%. It is around 2.5 times more than what India is happening. Are we very, very resistant? Are our immune system is very good? If we are questioning in the other side, we are not nutritionally very, very enriched people. Our children are not getting very nice food. Still we are surviving, while other people in Spain, America, in Brazil and UK, they are dying very fast. What is the reason? We have to think about that. We have to do research about that and find out why those people have died so much. And the severity cases, for example, severe cases, it was when started in Europe and America, it was around 5 to 12 to 3%. But now if you globally, you will check, it is only 1%. That means the corona lethality is going down, but still there is lethal. In America, again, it has revived 2 days back. So we have to ask ourselves why it is happening so. How whether the virus is getting mutated or not, how it is, what is the rate of mutations. All these questions are research questions, very, very relevant questions. And everybody, starting from a virologist to a genomist to a protein biochemist, everybody has to be involved in answering those questions. For example, how it has given research on acceleration. Question raised, whether it has been synthesized or has come from Juan's seafood. If it is from one seafood market, then how much time it has taken to actually to get the genetic and to make human as its own host, and it started killing. Why it is too lethal than SARS and MERS. SARS came in 2002, MERS came in 2012, and 2020 our COVID is killing our human race, and very, very differently. How it enters the body and interaction have happened. What is the mutation rate? So all these questions, whether they druggable targets for therapy, target for vaccines, and these are the questions people are asking. And you must be very happy that factors today about SARS-CoV is SARS-CoV might binds to the primary target cells like enterocytes and pneumocytes as for the, but these are the actually taken from a laboratory which is Goose laboratory, who published papers in 2007 and 2008. Similar viruses have been working there. So our knowledge is not exactly on the COVID or not exactly the corona which is killing us, but a similar system like virus, and who actually interacts like this to enterocytes and pneumocytes. Then other target cells like COVID may be epithelial, renal tube epithelial cells, or kidney immune cells, and celebration. And last week actually there was a paper which is saying that COVID infection can actually accelerate diabetes. So such type of research has to be required. Then there is complete sequence of the 2000 COVID is available, but there is only one source. People have not validated whether the same sequence is there or not. If it is the same sequence, if it is the same coronavirus, then it is only 95% of similar to bad virus, and 88% to the another species of. Then the recombination is very high, it shows. Therefore, one has to be very careful. If it is so, then why this recombination can be stopped. Why it is so fast recombining? There is a paper of John Hopkins who says that recombination is not very different. The mutation is not very different. So one has to challenge those questions and ask again and redo these things, research, so that to find out exactly what is happening. Now the total publication within these 3 months, you can see 28,986, where the total clinical trial to find out the best drug, including the vaccines, are 2,427. If you go back to the March data, it is only 250 around, and now it has become 2,427. So mostly the vaccines are targeting the spike protein. Out of the 4 exposed proteins of the coronavirus, the spike protein is the most targeted, and drugs are mostly repurposed because whatever we have for influenza and other type of diseases, we are trying to find out whether the same COVID can die out of this drug, and vaccines are mostly against the spike protein. So greatest realization is that we are, as a researchers in India, are at par with the other countries as far as COVID-19 challenges. Bharat Biotech has already come up with a killed virus vaccine, which is it is going to start in 15 places for the clinical trial 1 and 2. Jadus Kadila just yesterday, one day before, yesterday on Friday, they have got a modified plasmid who got a clearance from the DCGI. Then Serum Institute from Oxford, Serum Institute and Oxford, both Serum Institute of India and Oxford, they have some weekend virus, which is adenovirus. They have some this coronavirus fragments on it, and they are also trying to initiate another vaccine. So 3 vaccines are in line, and already Remdesivir is being marketed by Cipla and Hetero. Glenmark already started marketing Favipiravir. CDRI and Mediges is already started clinical trial in KGMU and Ram Lohia and Era Medical College on Umifenavir, and PRI, that is Patanjali Research, already launched the Coronil, which is actually not against the virus but about the increasing the immunity so that candidates can resist not to get the infection. Then therefore I would like to tell all my biologist young friends to be a researcher in original and change your dreams. Whether you are male or a female, you are equally important for contributing into science. These are the 3 people you must be knowing. The Samej is actually one of my mentors during my stay in John Hopkins from Johns Hopkins Schools of Medicine, and these 2 person, one from Harvard, and the other lady who is got in 2015 research Nobel Prize in artemisinin, and artemisinin is actually discovered in the city of Lucknow from CMAP and CDRA at the same time, but we are not so strong to put our case to get the Nobel, but she got it in artemisinin. So with these words, with all words of encouragement, I thank again to the organizers for giving me this chance to speak to the young biologists. Thank you so much.
Thank you, Dr. Rath, for enlightening our students. And you have told like how the students, what are the qualities there should be there in the researchers, young researchers, and also the research guides, and updating us with the COVID data. So now I request Dr. Ashutosh to introduce our last speaker of the day.